Good morning. Today's section topic is going to be about serological tests. So what are the meaning of serological tests? Serological, in the word serological, you can obtain the part which is sero for serology, and it gives you the term serum. So the serological tests are the tests which are performed on the patient's serum. And we're going to understand what are the meaning of serum. And for what aim is the serological tests performed? It's performed for the aim of the clinical diagnosis and identification of different diseases. So you're going to obtain the patient's serum and perform different serological tests, which we're going to get introduced to in today's section, for the aim of the diagnosis and identification of different diseases. So what are the difference between blood, serum, and plasma? So your blood contains many components, which are the red blood cells, the white blood cells, and the platelets. And for all of these components together, we call it the cellular portion of the blood. So the white blood cells, the red blood cells, and the platelets form the cellular portion of the blood. In addition to the plasma, and the plasma represents the fluid portion of the blood. And what are the plasma? Your plasma is made out of water, proteins, and clotting factors, like the fibrinogen. So here your blood is formed out of cellular portion and fluid portion. So as you can see, the plasma is a subset of your blood, containing water, proteins, and clotting factors. So what are the difference between the plasma and the serum? Simply, if you have your plasma, which contains the clotting factors, and you started to remove the clotting factors or the fibrinogen, which is one of the clotting factors, you will obtain your serum. So your serum is that plasma minus fibrinogen or minus the clotting factors. We call the serum the defibrinated plasma. So we can summarize the difference between the blood, serum, and plasma in this diagram. Here, as you can see, you have 3 circles. The biggest circle is the blood, and the circle in between is the plasma. So the plasma is a subset of the blood. It is the fluid portion of the blood. And the serum, which is the smallest circle in between, the smallest circle, it is a subset of the plasma, and it is the plasma minus clotting factors. So here your plasma, it's equal to your blood minus the cellular portion. So you removed the red blood cells, the white blood cells, and the platelets, and the remaining part is the plasma, which is the fluid portion. And here your serum equals to the plasma minus clotting factors. So in your lab, how are you going to obtain the serum in order to perform the serological tests? Simply, you're going to take a blood sample from the patient and put it in a test tube like this one, and then you're going to leave it to stand for like 30 minutes. And what's going to happen? Your platelets and your clotting factors will start to cause clotting of your blood. So here at the bottom you will find a blood clot, and here at the upper surface you will find a supernatant, which is your serum, which is a fluid portion containing no clotting factors, because the clotting factors have been used in the formation of the blood clot. After that, you're going to take your test tube and put it in the centrifuge in order to have a proper separation of the blood clot at the bottom and the serum at the upper part. So this is how you're going to obtain the serum. On the contrary, if you want to obtain plasma, how are you going to obtain the plasma from the blood sample obtained from the patient or taken from the patient? Simply, here you have a test tube containing the whole blood, and you have to add anticoagulant. Why you're adding anticoagulant? In order to prevent the coagulation of the blood. You don't want the blood to coagulate. You don't want your blood to clot. You don't want to use your clotting factors, right? So when you add anticoagulant, so you prevent the formation of the blood clot, and then you're going to take the tube and put it in the centrifuge, and after taking your tube out of the centrifuge, you will find it separated or fractionated into 3 layers. At the bottom, you have the red blood cells, and why it is at the bottom? Because it has the highest density. And in between, you will find a buffy coat containing the white blood cells and the platelets. And at the upper surface, you will find a supernatant, which is the plasma. And it contains the fibrinogen and the clotting factors, which haven't been used in the formation of a clot because you already added an anticoagulant. So this is just for your knowledge in order to understand how in the lab you can obtain a plasma from the blood or a serum from the blood. My main concern that you know that the serum is the plasma minus clotting factors. So one more time, here you have your patient. So you'll start to withdraw a blood sample from him, place it in a test tube, and you want to obtain from him the plasma. So the first thing you're going to do is that you're going to add anticoagulant. In order to avoid the coagulation of the blood, you don't want the formation of a blood clot. So after adding your anticoagulant, you're going to place your test tube in the centrifuge, and after taking it outside of the centrifuge, you will find it fractionated into 3 different layers. The erythrocytes, or your red blood cells at the bottom, the buffy coat containing the white blood cells and the platelets in between, and your plasma at the top. So here you obtained the plasma. And if you want to obtain the serum, you have to remove the clotting factors from the plasma in order to obtain the serum. And how are you going to do this? From the beginning, don't add anticoagulant and let your test tube to sediment so a blood clot is formed, and then add it to the centrifuge, and after taking it out of the centrifuge, you will have a blood clot here, and the supernatant will be the serum. So the main feature or the main principle of the serological tests is that it depends on the antigen-antibody interaction. To remind you, what is the meaning of antigen? The antigen is any foreign body entering your own body, and your immune system will start to respond or act against this antigen by the formation of a specific antibody acting against this antigen. And then the antigen will combine or bind to the antibody, forming antigen-antibody complex, and then a subsequent reaction is going to occur. So the main principle of the serological tests is the antigen-antibody interaction. So what I want you to know is that when you have a patient blood and you start to extract the serum from the blood, as we have learned in the previous slides, the serum contains the antibodies. So when you take the patient's serum, in order to perform different serological tests, it means you're looking for the presence or the absence of antibodies. So your serum contains the antibodies. So to test for the appearance or to test for the presence or absence of the antibodies in the serum, what do you have to add? You have to add your antigen. So you add antigen to the serum, which is suspected to contain antibodies, and if the antibodies are present, antigen-antibody interaction will happen, and if the antibodies are not present, no interaction will occur. So for the serological tests, you have 5 different types. So what are the 5 different types of serological tests? You have the agglutination reaction, the precipitation, the complement fixation, the hemagglutination and hemagglutination inhibition, the fluorescent antibody technique. In the practical part, we're going to study the agglutination reaction. And the remaining 4 types, you're going to study it in the theoretical part in the lectures. But my main concern is that you know that the whole 5 types depend on the antigen-antibody interaction. They differ only in the result of the antigen-antibody reaction. If the antigen-antibody reaction results in agglutination, which is coagulation or a very, very fine precipitated format, you call it agglutination reaction. So in the exam, I can ask you to enumerate the different types of serological tests. So you have to know the names, and as I told you, we're going to study the agglutination in the practical part and the remaining types in the theoretical part. So as we have previously agreed, your main principle of the serological tests is the formation of the antigen-antibody complex. So the first thing you're going to do is that you're going to obtain the patient's serum. And the patient's serum, you are looking for the presence or the absence of the antibodies. So in order to test for the presence or absence of the antibodies, you have to add antigens. So you add the antigens, you prepare it in the lab, okay? And if the antibodies are present in the patient's serum, the antigen will react with the antibody, forming antigen, and you can see your naked eye. So let us give an example. If you have a patient and you suspect that this patient might have an HIV infection, human immunodeficiency virus, so what are you going to do? You're going to obtain a serum sample from this patient, and you're going to add the HIV antigen, parts of the virus. Okay, so if this person is infected with the HIV virus, his serum will contain the antibodies. So the HIV antigen which you added will react with the antibodies which are present in the serum, and the result will be the agglutination. So this is how you're going to use the patient's serum in the clinical diagnosis and identification of diseases. Remember the main aim of the section? The first slide we have talked about. So if this person was not infected with HIV virus, there will be no antibodies in his serum, right? And when you add the HIV antigen to the serum, which contains no antibodies, will there be any agglutinin or agglutination reaction occurring? No. So if there is no agglutination, it means there is no infection. If there is agglutination reaction happening, it means the serum contains the antibodies and this patient is infected. So for the agglutination reaction, it can be done by either two methods. It could be done on a slide, or it could be done in a test tube. So that's why we have slide test and we have tube test. Slide test meaning that the test is performed on a slide, and tube test meaning that the test is performed in a test tube. For the slide test, we're going to study two things: the blood group determination and the Rh factor determination. And for the tube test, we're going to study the Vidal test. For your information, the Vidal test could also be performed on a slide. If you performed the Vidal test on a slide, it will be for the qualitative determination. But if you performed it in a test tube, it will be for the quantitative determination. And we're going to talk about the Vidal test next week. And for today, we're going to talk about the blood group determination and the Rh factor determination performed on a slide. So for the determination of the blood group, we have many different systems on which we can determine your blood group. But the main two systems, or the most two important systems, are called the Abo system, and this is the most important system, and comes after the Abo system is the Rh system, and this is the second important system for determining your blood group. So let's start with the Abo system. And by the way, this is an exam question. I can ask you what are the different systems for the determination of your blood group. So as we mentioned, there are many different systems, over 30 systems, but the most two important systems are the Abo system and the Rh system. So let's start with the Abo system. From your previous information and knowledge from high school, what are the different blood groups that you know? Yes, you know that you have the A blood group, the B blood group, the AB, and the O blood group. Why does these groups has the name of A, B, AB, O? This is because of the antigens which are expressed on the surface of the red blood cells, which means, so as you can see, for the blood type A, why is it called blood type A? Because if you looked at the surface of the red blood cells, you will find that the antigen which is expressed on the surface of the red blood cells is the A antigen. So from the name of the antigen expressed on the surface of the red blood cells, you have the name of the blood group. So for the blood type B, why is it called blood type B? Because on the surface of the red blood cells, you have the B antigen expressed on it. And for the blood type AB, why is it called AB? Because on the surface of the red blood cells, you have both antigens, antigen A and antigen B. And for the blood type O, and O reminds you with the 0, which means there is no antigen A or antigen B expressed on the surface of the red blood cells. Do you see antigen A or antigen B? No, there are no antigens A or B. And also O comes from a German word which is ohne, and ohne in German means without. So it means that the red blood cells are without the A antigen and without the B antigen. So you have two different antigens which might be expressed on the surface of the red blood cells, either antigen A or antigen B. If you have antigen A only, your blood type will be A. And if you have antigen B only, your blood type will be blood type B. And if you have both antigens, antigen A and antigen B, your blood type will be AB. And if you don't have antigen A, neither antigen B, your blood type will be O. And as you have two antigens, which are antigen A and antigen B, you will also have, yes, antibodies. And the antibodies which might be found are the anti-A, which is antibody against antigen A, and there is another antibody which is anti-B. Anti-B meaning antibody against antigen B. So where are these antibodies found? You tell me. Yes, the antibodies are found in the serum, right? We have mentioned that in the beginning of the section. So the antibodies are found in the serum, while the antigens are found on the surface of the red blood cells. So these antibodies are going to be found in which blood type? If you have blood type A, which is the red blood cells containing antigen A, do you think it will contain anti-A? Do you think you will contain the antibody against your self-antigen? Of course not. Logically, your body is not going to form antibody against your self-antigen. Since this is your self-antigen, which is not a foreign antigen, it's a part of your red blood cells, you're not going to have antibodies against it. But you are going to have anti-B, which is against antigen B. If your blood type is A, so you have antigen A, which is your self-antigen, but you don't have antigen B, so antigen B is something foreign. So your body contains anti-B. Also, if your blood type is B, so your self-antigen is the B antigen. So of course your body is not going to produce anti-B, which is against your self-antigen, but your body will be having the anti-A. Why does it have the anti-A? Because you don't have the antigen A expressed on the surface of the red blood cells. So the antigen A is a foreign antigen for your body. So that's why you have the anti-A. How about the blood type AB? It contains antigen A and antigen B. So antigen A and antigen B are both self-antigens. Do you think you'll have anti-A or anti-B against your self-antigens? No. So here you don't have any antibodies in your serum which are against your self-antigens. How about the blood type O? The blood type O contains neither antigen A nor antigen B. So here you don't have both antigens. So this means that you will have both antibodies. You will be having in your serum anti-A and anti-B, right? So here, to avoid any confusion, you know the name of the blood type from the antigens which are expressed on the surface. And you know the antibodies which are opposite to the name of the blood type. Which means if you are blood type A, your serum will contain the opposite of A is anti-B. And if your blood type is B, so your serum will contain anti-A. And if you are blood type AB, your serum will contain no antibodies. And if your blood type is O, your serum will contain both antibodies, which are anti-A and anti-B. So here, as we have said, that the blood group or the determination of the blood group, it consists mainly of two systems. What are the two systems? We said the first system is the ABO blood group system, and this is the most important system. Why does it have this importance? Because in the human blood transfusion, if there's someone who needs blood transfusion, he might made an accident and lost a lot of blood and needs blood to be transfused. In this case, you have to make sure that you are transfusing compatible blood, or a blood that matches his own blood. You cannot transfuse any blood type to any blood type. You must make sure that the donor blood type is compatible with the recipient blood type. Otherwise, if they are not matching, lysis of the donor red blood cells will occur. And if hemolysis of the red blood cells of the donor occur, degradation products will result from these lysis, and it will cause an overload on your spleen and your liver, and this might cause death. So this is the importance of knowing your blood group because of the cases of the blood transfusion. And the lysis of the red blood cells, or the hemolysis of the red blood cells, is called complement-mediated lysis, which means that the lysis of the red blood cells occur with the help of a protein which is called the complement protein. The complement protein is found in your blood on the formation of the antigen antibody complex, which is the antigen expressed on the red blood cell, and the antibody, which is against this antigen expressed on the red blood cell. On the binding of them and formation of antigen antibody complex, the lysis of the red blood cells will occur with the help of the complement protein found in the blood. So this is why it's called that the lysis is mediated with the complement protein. So here the ABO blood group system involves two antigens and two antibodies. What are these two antigens? Antigen A and antigen B. And what are these two antibodies? Antibody A and antibody B. And as we have said, where are these antigens expressed? Where can I find the antigen A or the antigen B? Yes, they are expressed on the surface of the red blood cells. And for the antibodies, where can I find them? I can find them in the serum. So here, the two antigens are antigen A and antigen B, and the two antibodies are antibody A and antibody B. The antigens are present on the red blood cells, expressed on the outer surface, and the antibodies are found in the serum. So regarding the antigen property of the blood, all human beings can be classified into four groups: blood group A, blood group B, blood group AB, and blood group O. And as we have said, the name of the blood group comes from the name of the antigen. Blood group A, meaning that the red blood cells contains or over its surface express the antigen A. And blood group B, over the red blood cells, you have antigen B. Blood group AB, you have antigen A and B. And blood group O reminds you of ONA, which means without A antigen and B antigen. So there is no antigen A, no antigen B. So here for the blood group B. I know you might find it annoying for repeating things over and over again. We're like refreshing our information, and we're going to add more on it in today's section. So here for the blood group B, we said that from the name of the group, we know the name of the antigens. We have the B antigens expressed on the surface of the red blood cell. So you tell me, what are the antibodies which will be found in the serum? Could it have anti-B, No, there is no anti-B. There will be anti-A. So here the serum contains antibodies which are anti-A. And as you know that the antibody has Y-shaped, right? So here, as you can see, we are going to detect the blood type from the diagrams which we have. As you can see here on the red blood cell, on the outer surface, you have antigen A. So what is the blood type? You have antigen A, so the blood type is going to be, from the name of the antigen, it's blood type A. And what is the antibody, which is going to be in the serum? It's anti-B. For antigen B, which is expressed on the surface, so you're going to have blood type B, and what's in the plasma or in the serum for the antibody, it's going to be anti-A. And for antigens A and B, so the blood type is going to be blood type AB, and for the antibody, it's going to be no antibodies because it has both antigens. And here there is neither A or B, so the blood type is going to be O, and the antibodies are antibodies A and antibodies B, right? Okay, so here is a very, very important information. We can classify the antibodies into different types. Your antibody could be monomer, which means you have like a singlet Y shape, right? There are different our other antibodies which are dimer. Here you have two Y shape. And there are antibodies which are of the pentamer type. Here you have 5Y shaped. So the different types of the antibodies, monomer, dimer, or pentamer. So an example of the monomer antibodies are the IgD. Here IG stands for immunoglobulin. So it's immunoglobulin D. IgE is immunoglobulin E. IgG is immunoglobulin G, and so on. For the dimer, you have the IgA, and for the pentamer, you have the IgM. In order to help you to memorize it, I will give you an acronym of Mediated. Mediated is an Arabic name, which is well known in the Arabic countries. It's a male name, actually. So these letters are going to remind you of the different types of the antibodies. Your antibody could be IgM, and this is a pentamer type. Your antibody could be of the IgA type, and this is a dimer type. And your antibody for the GED, it could be IgG or IgE or IgD, and this is monomer. So here are the different antibodies summarized in the word Mediated. What we're going to focus on, the IgM and the IgG, which is the pentamer and the monomer. An example of the IgM antibody is the antibody A and the antibody B. So the antibody A and the antibody B are of the IgM type, which is a pentamer antibody. So it means it's very large in size. Okay, and please make many stars here, because later on we're going to know having a large size and being a pentamer antibody, what did this information is going to be useful in the next information mentioned. So this is the first property of the anti-A antibodies and the anti-B antibodies, that they are pentamers of the IgM type. The second property of the anti-A and the anti-B antibody is that they are produced in the first years of life, which means if you are of a blood group A, so your serum contains anti-B, right? As you have known from the previous sections, in order or for your body to produce the anti-B, it must see and witness the B antigen, which is a foreign antigen. So here is an exceptional case. Your body is not going to wait to see or recognize the antigen B, which is foreign, in order to form the anti-B. No, your body will produce the anti-B in the first years of life without the recognition of the antigen B. When the scientists start to search how does that happen, how your body formed antibody against an antigen which your body hasn't seen before. How does that happen? They found out that antigen A and antigen B are carbohydrates in nature. And during your first years of life, you are exposed to different environmental factors like food, which is carb in nature, like microbe or many different microbes which might have like carbohydrates or polysaccharides expressed on the outer surface. So due to the similarity between the carbohydrate nature of the antigen A or antigen B and these environmental factors, this is called sensitization. Your body started to be sensitized against something which is similar in nature to the antigen A and the antigen B, and it will start to form the anti-A or the anti-B without the previous recognition of the corresponding antigen. Okay, so my main concern here, not to be confused, that you have to know the two important properties of the anti-A antibodies and the anti-B antibodies. What are the two main properties? The first property is that they are pentamer antibodies of the IgM type, so they are large in size. And the second property, that they are produced in your serum in the first years of life without the previous exposure to the corresponding antigen. If you are blood group A, your self-antigen is A, expressed on the red blood cells, and your serum contains anti-B. So we are done with the first system, which is the Abo system. The second system we're going to talk about for the determination of the blood group is the Rh blood group system. And this depends on the Rh factor, or we call it the rhesus factor, or we call it, a synonym of it is the D antigen. D antigen. So why do they give the D antigen the name of the Rh factor or the rhesus factor? Actually, because they first discovered the D antigen in a species of monkeys called the rhesus monkeys. So they named the D antigen after the species of monkeys which they first discovered the D antigen in. So, if the D antigen is expressed on the surface of the red blood cell, you will be Rh positive. And if the D antigen is not expressed on the surface of the red blood cell, you will be Rh negative. So here you are. If your blood had the antigen, you are Rh positive. And if your blood lacks the antigen, you are Rh negative. And Rh is your D antigen. So we previously know that we have 4 blood groups according to the ABO blood group system classification, which is A, B, AB, and O. So now after adding the Rh system classification, your A blood group might be A positive or A negative, which means you might have the D antigen or you might not have it. And for the B blood group, it might be B positive or B negative. And for the AB, it could be AB positive, AB negative. The same goes for the O, it could be O positive or O negative. So now you have 8 different blood group system, 8 different blood group types according to the 2 different classification systems, which are the ABO system and the Rh system. So here, as we can see, on the surface of the red blood cell, we have the Rh antigen, which is the D antigen. So this person is Rh positive. And here, on the surface of the red blood cell, there is no Rh antigen or no D antigen, so he is Rh negative. And for every antigen, there is an antibody. So, of course, there is an Rh antibody or D antibody, anti-D. Who will be having the anti-D, the Rh positive or the Rh negative? Of course, the Rh negative will be having the anti-D. Why? Because the Rh positive has the antigen, so it's a self-antigen, so of course it will not produce antibody against its self-antigen. Okay, so here let's take an example. If we have a person whose blood group is AB positive, so you tell me, for the red blood cells, what are the antigens which will be expressed on the surface of the red blood cell of AB positive person? We said from the name of the blood group, we can know the antigens expressed, right? So it will have A antigen, B antigen, and also D antigen, right? So it has 3 different antigens expressed on the surface of the red blood cells. How about the antibodies? Will there be any antibodies in the serum? No, there will be no antibodies, no anti-A, no anti-B, no anti-D, because all of these are self-antigens and the body will not produce antibodies against the self-antigen. So here, as you can see, the 8 different blood groups according to the ABO classification system and the Rh classification system. So in this slide, we're going to talk about the 2 properties of the antigen or the anti-D. The anti-D, which is the antibody against the antigen D, or we call it the anti-Rh. What are the 2 important properties of it? The first property is that the anti-D or the anti-Rh is not formed during the first years of life, like we have mentioned in the anti-A and the anti-B. No, they are formed in your blood or in your serum when your body get introduced to the antigen D. So how your body is going to get exposed to antigen D, which is foreign for you if you are Rh negative. This occurs in 2 cases. Either during, it happened by mistake during the blood transfusion. You are an Rh negative person, okay, so you don't have the antigen and you still don't have the antibodies. So if you got transfused with a blood which is Rh positive, so your body will start to recognize the antigens, the D antigens expressed on the red blood cells of the donor. You will start to form anti-D. So here the anti-D are formed when you got introduced to the antigen, either by mistake during blood transfusion or during pregnancy. the placental exposure during pregnancy, and we will understand this more in the next slides. So this is the first property, that the Ntrh are formed during blood transfusion, which happened by mistake or during pregnancy. And the second property of the Ntrh is that it is of the IgG type, which is a monomer, which means it's very small in size. Do you remember the Nta and the Ntb? Which type was it? Yes, it was of the IgM type, being a pentamer. So here is a comparison between the Ntrh or the Ntd compared with the Nta and the Ntb. You have two properties which you shall study it and know it, that the Nta and the Ntb are formed during the first years of life, while the Ntrh or the Ntd will be formed when your body gets exposed to the D antigen, and this occurs either by a mistake during a blood transfusion or during pregnancy. And for the second property, that the Nta and the Ntb are pentamer type, which is IgM, very large in size, while the Ntrh is of the monomer type, IgG, which is small in size. So for the blood group test, how are you going to determine your blood group type? Simply, you are going to sterilize your finger with an alcohol swab, and then with a sterile lancet, you're going to prick your finger in order to obtain blood. And on a slide, as we said, this is a slide test performed on a slide, you're going to add 3 different drops from the same person. This blood drop contains red blood cells on which there might be antigens expressed. What are the 3 different probable antigens which might be expressed? The antigen A, the antigen B, and the antigen D. You don't know which antigens are present. You're going to test for the presence of these 3 antigens. That's why you're adding 3 blood drops. So in order to test for the presence of these antigens, you are going to add antibodies. So what are the antibodies added? You're going to add Nta antibody, which is the blue one, and you're going to add Ntb antibody, which is the yellow one, and you're going to add Ntd antibody, which is the transparent one. So here you are adding the Nta to your blood drop, and here you're adding the Ntb, and here you are adding the Ntd, which is the Ntrh. So here, your first drop, you found out that agglutination occurs. There is an agglutinin, there is coagulation. So that means that the antigen A was present and it reacted with the antibody A. So here you have antigen A expressed on the surface of the red blood cell. And here on the second drop, there is no agglutination, it's totally clear. So the antibody B, which is added, found no antigen B, so no agglutination occurred. So there is no antigen B. And here, on adding antibody D, agglutination occurs. So it means that there is antigen D expressed on the surface, and it reacted with the antibody D, and it caused the agglutination to occur. So this person blood type is A positive, right? Another example here, you found that there is agglutination here and here, right? And here there is no agglutination. So here it means there is antigen there is antigen B, which reacted with antibody B, and there is antigen D, which reacted with antibody D. So this person is B positive. And here you can find that for another person that there is agglutination in the 3 drops. So it means he has antigen A, he has antigen B, He has antigen D. He has the 3 antigens, so he is AB positive. For the last person, there is no agglutinations in any drop. Here it's clear, clear, and clear, so this person is O negative. There is no antigens expressed on the surface of the red blood cells. So what is the importance of knowing your blood group type? Actually, there are two importance. The first importance is for the transfusion reactions on transferring a blood from a donor to a recipient. We said that the blood should be compatible, should be matching. And the second importance is in the pregnancy. So let's talk about the first importance. It tells you that no individual will ever receive a blood transfusion based on the Abo system alone. You have to put in your consideration the two systems, the Abo system in addition to the Rh system or the rhesus factor. So both of them are very, very important. And both systems are the most important compatibility factors to consider. So, when you transfer blood from donor to recipient, donor will give to recipient. The donor could donate whole blood. You can take whole blood, or he can donate packed RBCs. We take the blood and fractionate it and take from the blood only the RBCs, and we start to transfuse it to the recipient. Why RBCs specifically? Because, as you know, it contains the hemoglobin and it's very, very important for carrying the oxygen. So I will talk about the case of transfusing packed RBCs. So when you transfuse packed RBCs, you are transfusing red blood cells with antigens expressed on the surface of the RBCs. So you look for the donor, you look for the antigens of the donor, right? And for the recipient, you're going to look at the serum of the recipient. So in order to determine whether there is compatibility or not, you're going to look for the antigens of the donor, the antigens transferred or transfused, because you are transfusing packed RBCs, and for the recipient you're going to look for the serum. And of course, in order to say that both blood types are compatible, there shouldn't be any antigens in the donor which is acting against or will bind with the antibodies which are found in the serum of the recipient. So let's take an example. If we said that there is someone who is going to donate blood to another person, and the donor blood is of blood group A positive, and the recipient blood is going to be B positive, would that be compatible or not? And this is an exam question. I can give you any two different blood types and ask you whether they are compatible or not compatible. So for the donor, here we said we are transfusing packed RBCs, so we are going to look for the antigens. So what antigens are we transferring or transfusing? The A antigen, the B antigen, and the D antigen. Right? So here you have 3 antigen being transfused. And for the recipient, we said we're going to look for or to look at the serum. His blood group is B positive. So what are the antibodies which are in the serum? Yes, he has anti-A. Does he has anti-B? No, because he has a self-antigen. Does he has anti-D? No, because he has the self-antigen of the antigen, so he only has anti-A. So as you can see, you are transferring antigen E to a person whose serum contains antibody A. So what's going to happen? Antigen-antibody reaction, right? And hemolysis will occur, lysis of the Rbcs, and this is called hemolytic transfusion reaction or complement-mediated lysis. Lysis of the Rbcs is mediated with a complement protein. And of course, the lysis will occur to the donor red blood cells, and this will lead to the formation of degradation products, which will cause an overload on your liver and on your spleen, and that might lead to this. So both bloods are not compatible. So let's take another example. If the donor blood is group B positive and the recipient is group AB negative, are they compatible or not? So if you have this in your exam, you have to answer stepwise. For the donor, you're going to look for the antigens, which are expressed on the surface of the packed RBCs. So here you have the B antigen and the D antigen. And for the recipient, you're going to look for the serum containing the antibodies. So here, do we have antibody A? No. Do we have antibody B? No. Do you have antibody D? Yes, but after the exposure of your body to antigen D, which means if this is the first time of transfusing Rh positive or B positive to AB negative, so the body hasn't formed the antibody D yet. So the antigen D will enter, and there is no antibody D formed yet. This is the first time. And it will start to form antibody D, and it will take time. So the first transfusion will pass safely. So the first transfusion will pass safely. But if this occurs another time, a second transfusion, the antigen D entering your body will find the antibody D ready-made with large amount, so it will start to bind with the antigen D, and here antigen-antibody reaction occurs, and complement-mediated lysis or hemolytic transfusion reaction occurs after the second blood transfusion. So one more time, when you transfuse an Rh positive person to an Rh negative person, if this is the first time, the body of the Rh negative person hasn't formed yet the antibody. So for the first time, the immune system will start to recognize the foreign antigen D and will start to form antibody D, and this takes time. So the first transfusion will pass safely. But for the second transfusion, if this mistake is repeated one more time, the antigen D, which is re-entering your body, will find the antibody D ready-made with large amount, so it will bind with the antigen D, and hemolysis of the red blood cells will occur. So here is a table which is summarizing the 8 different blood groups for the donor and the recipient. And if we look here, we will find that the donor, which is the O negative, can donate to all of the other blood types, right? And here in the recipient, we will find that the AB positive can receive from all other blood groups, right? So here we call the O antigen, the O negative blood type is the universal donor. It can donate to all other blood groups. And the AB positive is called the universal recipient because it can receive from all other blood groups. So the question is why the O negative is a universal donor and why the AB positive is universal recipient. We said that for the donor, we are donating here packed RBCs, so we are looking for the antigens, right? The O negative, does it has any antigen? No, it has no A antigen, it has no B antigens, and it has no D antigens. So since it has no antigens, so there will be no problem. So that's why O negative is a universal donor. While for the AB positive, why it is the universal recipient, we said that for the recipient, for the receiver, we look for the antibodies which are found in the serum. For the AB positive, does it has any antibodies? No, there is no anti-A, no anti-B, and no anti-D. So it has no antibodies in the serum. So whatever antigens you are transferring, there will be no corresponding antibodies. So that's why the AB positive is a universal recipient. So here, from this diagram, we have the 4 different blood groups. As we have mentioned, the O blood group, and specifically the O negative, is a universal donor. So as you can see here, it can donate to the A, to the AB, and to the B. And we said that the AB positive is a universal recipient. So you will find all arrows going to the AB, as it can receive from all other blood groups. So here is the universal donor is blood group O negative, and the universal acceptor is AB positive. And please remember, this is the case on donating packed RBCs. Because if you are not transferring packed RBCs, but you are transferring whole blood products, it means that you are transferring RBCs plus plasma or plus serum, right? And the serum contains antibodies, right? So you are transferring antigens and antibodies. So if you are transferring whole blood products, you have to transfer the same blood type of the donor to the same blood type of the recipient, which means the A positive to the A positive, the B positive to the B positive, the O negative to the O negative, the same blood type for the donor and the recipient. But what we have mentioned before for the universal donor and the universal recipient, it applies only to the packed RBCs, because you're only transferring RBCs, no serum transferred. So we are done with the first importance of knowing your blood group type, which is the human blood transfusion reaction. The second importance is during the pregnancy. During the pregnancy, a pathogenic condition might occur, which is called the hemolytic disease of the newborn, or there is another synonym of it, which is the erythroblastosis fetalis, or the Rhesus disease, or the Rh disease. All of these are synonyms. So this condition or this disease occurs as a result of the incompatibility between the blood types of the mother and the fetus, especially the Rh factor. And here, erythroblastosis, erythroblast, it is the red blood cells, and osis, it means a diseased condition. So from the name, you can understand that there is a diseased condition in the RBCs, which is the lysis of the RBCs. And the lysis of the RBCs will occur due to the incompatibility between the blood types of the mother and the fetus. So this problem happens when the mother is Rh negative and the fetus is Rh positive. During the first pregnancy, things go smoothly. No problem happens. But during the delivery, when the placenta detach, mixing of the blood of the mother and the fetus occurs. So some of the Rh positive RBCs is mixed with the blood of the mother. So the mother body will get exposed to the RBCs of the fetus with the D antigen expressed on its surface. So the mother will start to form Ntrh antibodies, right? So during the second pregnancy, when the Rh negative mother is pregnant with an Rh positive fetus, what will happen? As we have said that the Ntrh antibodies are of the IgG type, right? And we said that this is monomer antibody. So it is of a small size and it can cross the placenta. So the IgG antibodies will start to cross the placenta, reaching the fetus with the D antigen or the Rh factor expressed on the surface of the RBCs, thus causing hemolysis of the RBCs of the fetus. And this is caused the hemolytic disease of the newborn or the erythroblastosis fetalis. So in order to avoid the occurrence of this disease, during the first pregnancy, on the delivery time, when the placenta detach and mixing of the blood of the mother and the fetus occurs, the body will get exposed to the Rh antigen or the D antigen. In order to avoid the formation of the antibodies by the body of the mother, you have to give her antibodies which are ready-made. Anti-D antibodies which are ready-made, and this is called the Ntrh injection. I'm sure that you have heard about it, that when there is an Rh negative mother pregnant with Rh positive fetus, they have to give her Anti-Rh injection. What is the Anti-Rh injection? It is the Anti-Rh antibodies or the Anti-D antibodies which are ready-made. You give it to the mother immediately after the pregnancy. It should be within 72 hours of the delivery time, in order to prevent the body from recognizing the RBCs of the fetus with the expressed H antigen, with the expressed D antigen. So the injected Anti-Rh will start to neutralize the Rh antigens and break it down, and the body will not recognize the RBCs of the fetus, and it will not form anti-D antibodies. And of course, this injection, the mother should take it after every pregnancy, in order to avoid the occurrence of the hemolytic disease of the newborn in the subsequent pregnancy. This is what we have said. So if you haven't given the Anti-Rh injection after the first pregnancy, the second delivered baby will have the erythroblastosis fetalis. So for the fetus, which is not yet born, what are the symptoms and the signs? The symptoms and the signs are enlarged liver and spleen, and this occurs due to the hemolysis of the red blood cells and the degradation products, which result in cause an overload on the liver and the spleen. And of course, enlarged heart, because no enough blood is reaching the heart due to its hemolysis. So as a result, the heart will increase in size. If the fetus survived and it was born, the signs and symptoms in the newborn will be anemia, of course, because of the hemolysis of the RBCs, so there is no hemoglobin, jaundice, and it occurs due to the degradation of the RBCs, resulting in a product called the bilirubin, and as you know, it will cause the yellow coloration of the skin and the eyes. And enlargement of the liver and the spleen due to the degradation products and the overload on the body, severe edema, and dyspnea, or difficulty in the breathing. Why does that happen? Due to the degradation of the RBCs, the degradation of the hemoglobin, which is the carrier of your oxygen. So how could you prevent the occurrence of such a disease? As we have said, you have to inject the mother with the anti-D antibodies or the anti-Rh antibodies. It's called raw immune globulin within 72 hours of the pregnancy. In such case, the ready-made antibodies, which you're going to inject the mother with, will start to neutralize and break down the antigens, the D antigens of the RBCs of the fetus, and the body will not recognize the antigens and will not form antibodies against it. And you have to do this after each pregnancy. So now we're going to talk about a phenomenon which is called the Bombay phenotype. And it holds the name of Bombay because it was firstly discovered in Bombay in India. Before explaining this phenotype, I want to explain something to you. Here if you have RBCs, on the surface of the RBC, there is an antigen which is called the H antigen. Have we mentioned the H antigen before? No. We have mentioned before the E antigen, the D antigen, and the B antigen, right? So what is the H antigen? The H antigen is the precursor or the starting material for the A and the B antigen. It's the building block of the A or the B antigen. So for a person whose blood group is A, the H antigen will start to form the A antigen. So it will has H antigen expressed on the surface of the RBC in addition to the A antigen. For someone whose blood group is B, on the RBCs on its surface you will find the H antigen, which is the precursor or the building block, in addition to the B antigen. Okay, for someone who is O blood type, you will find on the surface of the RBC the H antigen only. So here the precursor or the starting material of the A and the B antigen will remain as it is. It will not be converted to the A or to the B antigen. And for a person whose blood type is AB, you will find that on the surface of the RBC, the H antigen will remain as it is, and also part of it will be converted to the A antigen, and part of it will be converted to the B antigen. So you will have the A, B, and the H antigen. So here in the four blood types, A, B, O, and AB, it's a must that you find the H antigen, which is the precursor of the A or the B antigen. For the Bombay phenotype, there is no H antigen. You don't have H antigen, and this is a rare case. So, if you don't have H antigen, do you expect having A antigen or B antigen? No. Since you don't have the precursor, you don't have the building block, you will not be having the A or the B antigen. So here, the person who is of a Bombay phenotype will look like the O blood type, but it's an O blood type without the H antigen, meaning that, as we have previously mentioned, that the O blood type on the surface of the RBC there is H antigen, and the H antigen will not be converted into A or B antigen, right? While for the Bombay phenotype, there will be no H antigen. The precursor is not found. Okay, so we can see that the Bombay phenotype is an O blood type without the H antigen. So here it tells you individuals with a rare Bombay phenotype, we give them the symbol of HH, don't express antigen H on the red blood cells. as H antigen serves as precursor for producing A and B antigens. So the absence of the H antigen means individuals don't have A or B antigens as well. They are similar to the O blood group, but what is the difference between them and the O blood group? That they don't have the H antigen, but the O blood group has the H antigen. So here, however, unlike O group, the H antigen is absent. So the individuals produce antibodies to antigen H, as well as to A and B antigens. So the Bombay phenotype in his serum, he will have the anti-A, the anti-B, and the anti-H. So in this case, if you tried to donate O blood type to a Bombay phenotype, what do you expect to happen? You have O blood type, or let's say O negative. We said it's the universal donor, and donating to the Bombay phenotype, donating HH. What's going to happen? For the universal donor, we're going to look for the antigens. Is there any antigens for the O negative? Yes, there is the H antigen. And for the HH, the recipient, we're going to look for the antibodies in the serum. If there are any antibodies in the serum, yes, there is the anti-A, the anti-B, and the anti-H. So here you will find that your universal donor, which is the O negative, cannot donate to the Bombay phenotype. So the Bombay phenotype can only receive blood from a Bombay phenotype as well. Another topic we're going to mention is the disease association, which means we are associating the blood type with certain diseases. This is not based on any scientific evidence, it's only based on surveys and statistics, so it's not an accurate thing. So here it tells you that the O group, compared to the non-O group individuals, they have 14% reduced risk of the different carcinoma, squamous cell carcinoma, basal cell carcinoma, pancreatic cancer, and so on. And for the B blood type, they has an increased risk of ovarian cancer. And for the A group, it's more common, they are more common to have the gastric cancer. So don't worry if you are in the A group or B group. All of this is based on surveys and statistics, no scientific evidence for this information. Another information is the subgroups. As we have known, we have the A group, the B group, the AB, and the O. There are subgroups below the blood type. For example, for the A blood type, it contains about 20 subgroups. You have A1, A2, A3, A4 until A20. And A1 and A2 are the most common. And A1 makes about 80%, and the remaining is for A2. So what's known that these two subgroups are interchangeable, they are compatible. But some complications can occur in rare cases. So if a blood group A is donating to a blood group A, the normal case or the known thing is that, of course, they are compatible. But in some rare conditions, if certain complications started to arise, in this situation, you have to attribute it to the subgroups. The problem will be coming from the subgroups. Someone might be A1, the other might be A2, and it might cause you a problem. But generally speaking, they are interchangeable, they are compatible, and in certain rare cases, certain complications might occur. So the last thing we're going to mention is the conjectures. What is the meaning of conjectures? It is the association of the different personality types and traits with the different blood groups. And actually, this is very popular in Japan. So, for example, they say that blood group O is associated with the perfectase, and blood group A2, they have the highest IQs. Of course, this has no scientific basis or scientific evidence. It's all based on surveys and statistics, so these information are not accurate. So before ending today's section, I have a question for you, and the first person who's going to answer it correctly and post the answer in a comment in the Microsoft Teams in our channel, he or she will be rewarded next week in our section. So the question is, if you have a pregnant mother and the mother blood group is A positive, and she's pregnant in a baby, so B for baby, whose blood type is B positive, would there be any problem or incompatibility problem between the mother and the fetus? Please answer with yes or no with justification. So thank you for your time, and please, if you have any questions in today's section, don't hesitate to contact me, and have a wonderful day. Thank you.